Thank you very much. Good morning, everybody. And my role is twofold. It's to show you how important diagnostics are in biopreparedness, and to show you how difficult it is to prepare with diagnostics and how we really need partnerships in this sphere. I think there was some mention before about how it requires less money and shorter time to develop diagnostics. It's true, but we have less money and we have less time to be able to do this. So everything is in proportion, everything is relative when you talk about diagnostics. So let's talk about a distant history lesson. Okay, those who can't remember the past are condemned to repeat it. And when the situation was manageable, it was neglected, and now it is thoroughly out of hand. We apply too late the remedies which might have affected a cure. So you can talk about Ebola, you can talk about Zika, you can really apply this to almost any epidemic. We come too late, we're unprepared, and it happens over and over again. If you look at Ebola, we've talked a lot about Ebola. On the right is a picture from the clinic where we did our diagnostic clinical trial in Guinea of our BioThread E assay. But essentially, the patients and the healthcare workers were crying for diagnostics. Vaccines would have helped, therapies would have helped, but they were crying out for diagnostics. If you look at Zika today, these are snapshots of the lay press from recently, the past 2 weeks. The people, the populations, the governments are crying out for diagnostics. Yes, we need vaccines. Yes, we need therapeutics. But right now, the crisis is in diagnostics. So additional recent history lessons, obviously Ebola and Zika. H1N1 taught us lessons. MERS-CoV, we're still not sure what's going to happen with MERS-CoV, whether it will spread beyond the Middle East and Korea. XDR-TB is already spreading. You heard a little bit about that. To come, there are yellow fever epidemics occurring. Totally drug-resistant Acinetobacter is occurring all over India and China, and now in North America as well. Candida auris, I don't know if many of you have heard about this, but this is a yeast which is resistant to all 3 classes of antifungal drugs. There are no treatments available for this fungus. And artemisinin-resistant Plasmodium falciparum, which is obviously the cause of malaria. If this gets into Africa, there's going to be a huge problem. So what I'm not talking about today is I'm not talking about viral pathogens. I know everybody's been talking about viral pathogens and the WHO list of priorities, but let's be frank here. Every time there's an epidemic, we're surprised by which pathogen is the cause, right? We didn't expect it to be a Zika virus. We didn't expect Ebola to be as transmissible as it is. We're going to be surprised again. So I'm not talking about viral pathogens. For diagnostics, we have to be prepared for all the important pathogens, whether they're viral, bacterial, parasitic, or fungal. The next big epidemic could be a artemisinin quinine resistant malaria. Okay, we don't know. And we're always surprised. I am not talking about molecular testing. Everybody talks about primers and probes. Let's get molecular tests. But as we've seen with Zika, sometimes it's serology which is needed, not a molecular test. So I'm not talking about just molecular testing and nucleic acid amplification tests. I'm talking about all diagnostic assays which could be required. And that could be lateral flow antigen testing, that could be immunoassays, that could be nucleic acid testing. I'm not talking about emerging countries only. Because yes, the lessons that we've had have been emerging countries. Saudi Arabia is not an emerging country. Okay, Korea is not an emerging country. These have had to wrestle with pathogens that are significant. The United States is now wrestling with Zika. Okay, there are going to be other countries wrestling with Zika. They are not emerging countries. Yes, Guinea and Sierra Leone had major problems, and we're going to see other problems in emerging countries. There's no question. But I'm not just talking. This has to be a global preparedness. I am not talking about hospital labs or clinics, because as we saw from Ebola, sometimes the best test needs to be used in the bush, under a tree, in a clinic, okay, at the side of a patient, in a bed of an area that has no lab whatsoever. So it's great to talk about labs, but not only labs. So what I will be talking about is what kind of diagnostics do we need for emerging pathogens? What are the major obstacles in developing such diagnostics? What can we do in advance to better be prepared? Because this is about biopreparedness. How does it work today? Today, to develop a diagnostic, all right, it is complex. It is not so easy. Everybody thinks that developing diagnostics are easy. Being in a diagnostic company, it is not easy. However, it is quite uncoordinated today, and there are 2 major pathways. The top pathway is what I call the academia public health pathway, and the bottom part is the industry pathway. So we all start at the same. So we start with recognizing the need on the left, and then we have to look and see whether there's actually going to be an outbreak or not, et cetera, et cetera. At bioMerieux, we have a team called DOIT, Diagnostic Outbreak Intervention Team, which takes these veilles scientifiques, these watches, we look for emerging pathogens, and then we see, is this going to spread? Do we need to develop a diagnostic? What are we going to do? Do we need a serology? Do we need a nucleic acid test, et cetera, et cetera? Should we act on it now? Where's the budget going to come from? Where are the people going to come from, et cetera, et cetera? We did this for Ebola. We did this for Zika, et cetera, et cetera. We all start with the same thing. But then after that, it splits. Academia and CDC, EVA, Pasteur Institute, all these networks, they create tests pretty quickly, but they're for a very limited use. They use them locally, they hand them out to reference labs, and that's where it stops. They do not have the capacity to industrialize, they do not have the capacity to deploy, they do not have the capacity to distribute globally. So it stops at these reference labs and selected areas where they distribute. Industry then has to go on a separate pathway through regulatory, industrialization, et cetera, which is very complex and is much more long and costly, and we have to take decisions that are based on market analysis and whether we're going to be distributing in which countries, et cetera, return on investment, NPV, all these things that industry does and that we have to do before we market a product. So it's a much more difficult process. An academic lab can develop a nucleic acid test in about a week, okay, 2 weeks. We can develop a test that quickly too, but to develop a test that's ready for the market does not take 2 weeks. All right, there's a huge difference there. So what kind of diagnostics do we need? Let's look at some examples. I'm an epidemiologist. Epidemiologist, if there's any in the room, our answer always is, it all depends. Whatever the question is, the answer is, well, it all depends. All right? And it's the same thing here. What diagnostic do you need? The answer always is, it all depends. Let's look at some examples. Ebola was a disease similar to other febrile illnesses. It looked just like every other fever. It was symptomatic in almost all the cases, and we needed a diagnostic for acute disease. There was a high mortality rate with a high transmission rate. Therefore, we didn't want to mix Ebola positive and Ebola negative patients because the Ebola negative patients would get Ebola and they would die. We wanted to minimize invasive sampling because the healthcare workers were very afraid of the patients, and rightly so. So they didn't want to take a lot of blood sampling and a lot of needle use. Many patients died prior to medical care, so there had to be a diagnostics for bodies that were lying in the street. All right, so they had to be validated on post-mortem specimens like saliva, and people were taking unvalidated tests and using them on saliva samples because they had nothing else, because they had bodies lying outside the clinics. The lab facilities in those countries were very basic, okay, that had to be remembered, in Sierra Leone, Guinea. So the medical needs were a rapid, easy to use, easy to read assay on urine, saliva, or blood with a high sensitivity, we didn't want to miss any cases, and a high specificity, we didn't want to overdiagnose and place those people into the Ebola areas. Okay? For that, it was obviously a nucleic acid test that was required, or a very highly sensitive lateral flow immunoassay test, which we had problems developing. Everybody had problems developing because of the sensitivity. Also, easy, rapid diagnostics for other similar diseases. Why other similar diseases? And I'll give you an example. This is an example of the EVD treatment unit of the British Defence Medical Services. They had an outbreak of Shigella in their healthcare workers at the same time in their Ebola unit. So they didn't know when people came in with fever and diarrhea and they were bleeding from their stool whether they had Ebola or whether they had Shigella. If they made a mistake and put a nurse with Shigella in with the Ebola patients, they would get Ebola and die, likely. All right? They didn't want to mix up, so they needed rapid diagnostics for Shigella, and they wound up using our BioFire FilmArray GI test, which is a 1-hour test for GI pathogens, and that helped them to delineate who was who when they walked in with fever among the healthcare workers and patients as well, but mostly among the healthcare workers. So they needed other diagnostics to help them in the Ebola clinic to handle Ebola. Okay, it sounds bizarre, but that's what it was. So that's what they were doing. They were doing a whole panel of tests. And this is their publication if you want to read it. Example number 2 is Zika virus. That's a different need for testing. It's a disease similar to dengue and chikungunya, which are endemic in the same countries where Zika is, and in most cases is asymptomatic, very different from Ebola. And most patients don't come to medical care. When you go to Brazil and you see how many pregnant women get Zika, most of them never go to see a doctor. There's no money in the healthcare system. The low mortality rate, a high complication rate in a certain subset of people, okay, pregnant women. The sample type is not an issue, but blood and urine seems okay. Few patients die of Zika. You don't need a post-mortem test. Zika is transmitted by transfusion of blood products, and that's become a huge problem in those areas, which is not the same for Ebola. Lab facilities in those areas are very variable. In Brazil, it's pretty decent. In other countries, it's not. In the United States, it's very high level, etc. So what are the medical needs? An assay which is able to differentiate susceptible from non-susceptible women and able to detect asymptomatic infection, et cetera, and to differentiate from dengue and chikungunya. Obviously, it's an immunoassay that's required, not a nucleic acid test for this population, all right, with some kind of point-of-care or mobile type of assay. A high-sensitivity, high-throughput assay for screening blood donors is required, or we're going to start transfusing Zika into patients. And so obviously there's a highly automated nucleic acid test that's required, and sensitive screening for diagnosing symptomatic pregnant women, obviously a nucleic acid test, but that's the third priority, the way we see it for diagnostics. If you look at MERS-CoV, it's totally different. I'm not going to go through the whole thing, but essentially we needed an assay which is able to diagnose acute disease from an invasive specimen. When we looked to see diagnostic assays, they only had bronchoalveolar lavages because those were the only patients that were tested. Those were the ones in intensive care unit. The rest of the patients with mildly symptomatic disease, they didn't care about MERS-CoV. So you had to have a good test that was validated on BAL, very different. You also needed a zoonotic test because there was the camels involved and they had to know which camels were colonized and which ones were carrying. So there was an assay that was required for BAL, which needed to be validated on BAL, and an immunoassay for seroprevalence studies of camels as well as of humans, a totally different validation process that's required. So what are the major obstacles to deployment? The major obstacles are, number one, when we have conference calls with the CDC and we talk about what's the difficulties in making a MERS-CoV diagnostic, okay, the lack of standardized and well-characterized biobanks of clinical specimens, organisms, interfering substances like other assays. This is the major obstacle that we all face. The lack of raw materials like monoclonal antibodies, purified immunoreactive antigens, and primers and probes. A dearth of BSL-3 and BSL-4 labs that can be worked in to create the diagnostics. We had to wait 4 months to get access to the BSL-4 lab in order to test our Ebola test and inactivation with our buffer, et cetera, et cetera, et cetera. I mean, you have to line up to get space in a BSL-4 lab. The lack of point-of-care platforms. We just don't have point-of-care platforms for immunoassays and nucleic acid testing. As much as we all talk about it, I think there's 40 different SMEs that are now working on point-of-care nucleic acid, and we really don't have them yet. Difficulty in conducting clinical trials in real patients. We had an Ebola test available within 3 weeks of the, because we already had the primers, probes, et cetera, and we already had the BioFire platform. It then took us 4 months to get regulatory and ethics approval in Guinea to get the study underway. We were ready with our team and our equipment and our tests and the clinic and everybody all prepared, and then it took 4 months for ethics approval. It's ridiculous. In a time at the peak of the epidemic when everybody was screaming for diagnostics, 4 months for ethics approval in a country with the government working on it, and unfortunately the government being one of the major obstacles. Regulatory approval was problems. Supply chain issues. We have big supply chain issues with diagnostics. How do you deliver them? They have to be maintained, quality control, et cetera, et cetera. There's education involved, connectivity, and all that. Education of the people that are doing it. So there's a lot of preparations that can be done in advance. Among that whole list, okay, all of these can be done in advance. In a biopreparedness consortium or preparatory group, these can all be done in advance. Okay, facilitating regulatory approval, facilitating the availability of biobank specimens, et cetera. Okay, so in conclusion, the diagnostics for addressing emerging pathogens will depend on the respective needs. There's not just one nucleic acid test that's required that we need to prepare for. Biopreparedness needs international cooperation and academic private partner collaborations. Public-private, such as the one that I believe that Pasteur and FIND are trying to initiate, that have gone through the R&D application process with WHO. Those kinds of consortia would really facilitate this kind of thing. Deployment of diagnostics is more than just developing a test. It's regulatory approval, manufacturing capacity, supply chain issues, education and quality control, reimbursement and payment issues. It's a big problem. Thank you.